Myself, I am Dr. Rangarao, working as Associate Professor in the Department of Biotechnology. So I am dealing biochemistry for second year students. So topic is polysaccharides. Today we are going to discuss about the poly. So this topic covers about polysaccharides and then homopolysaccharides and then heteropolysaccharides, complex carbohydrates, structure of starch, glycogen, structure of dextran, cellulose, glycoproteins, glycose, aminoglycans, anomerism, isomerism, metarotation. So this topic covers this whole contents. Saccharides. So polysaccharides contain sugar units. So this sugar units linked to one unit to another unit. So and then these polysaccharides contain straight chain form and branch chains. So if you see here one sugar unit linked to another one, sometime the linkage alpha 1, 4, alpha 1, 6, alpha 1, 3. It depends on the molecule carbohydrates. So cellulose, example some of the polysaccharides, cellulose, chitin, peptide, glycan. So cellulose usually found in the plants. So and then the cellulose, if you want to break the cellulose, it is very tough. Okay, so most of the organisms unable to break the cellulose bonds. Then coming to chitin, so this chitin actually forms exoskeleton in insects and crustaceans, and this sugar units linked to amino acids and the coming to peptide glycan found in cell walls of bacteria. Polysaccharides are complex carbohydrates composed of several sugar units arranged in straight chain and then branched chain. The most of monosaccharides that appears as a parts of the polysaccharides, nothing but glucose, fructose, galactose and mannose. And polysaccharides are polymer of monosaccharides. They will form polymer, the branched chain or straight chain. Same type or different type. Classification of this polysaccharides, which depends on the biological role. So storage polysaccharides and then structural polysaccharides. Coming to storage polysaccharides, example starch and glycogen, whereas structural polysaccharides, cellulose and chitin. Classification of monosaccharides types, homoglycans and heteroglycans. And properties of polysaccharides, these polysaccharides don't have a sweet taste, insoluble in water, the molecular weight is very high, and it forms colloidal solution when heated with water. They don't exceed any of the properties of aldehyde or ketone groups. Homopolysaccharides, they contain monosaccharide units of a single type, example glycogen, starch, cellulose, chitin, and then polymer of alpha glucose is a starch. Starch actually contains alpha glucose units. This will form large chain, polymer of the alpha glucose, whereas cellulose contains polymer of beta glucose. Heteropolysaccharides, they possess two or more types of monosaccharides units. Example, heparin, this is the structure of heparin, and complex carbohydrates. They are made up of sugar molecules that are stretched together in a long chain. They are found in foods such as beans, whole grains and vegetables. Both simple and complex carbohydrates are turned to glucose in the body and are used as a energy. Keep the body fueled for an extended period of time. Glycogen is a homopolysaccharide. It gives glucose on complete hydrolysis. It is a glucon. Mainly, this glycogen is storage carbohydrate. Usually, it is in liver, muscles of animals. It is a similar structure to amylopectin. It is called animal starch. It is in the form of the powder, white powder dissolved in the water. This actually contains alpha 1, 4, alpha 1, 6 types of the linkages by glycosidic bond. The straight chain contains alpha 1, 4 glycosidic bond. The point of branch contains alpha 1, 6 glycosidic bond. This is the structure of glycogen. If you see here, the sugar units actually linked to one unit to another unit, alpha 1, 4 and then alpha 1, 6 when it is formed the branched chain. And starch is a homopolysaccharide. It is a non-sugar molecule. It yields glucose on complete hydrolysis, so it is a glucon. It is a main storage material in plants. Starch is insoluble and it is easily available for use by the plant. It is a white, amorphous, tasteless and soft substances. It is made up of glucose units. The glucose units are linked by alpha 1, 4 or alpha 1, 6 glycosidic bond. Starch consists two molecules. So one is amylose, it is contained 20%, and amylopectin, it is contained 80%. So amylose, it is made up of glucose units, alpha glucose molecule that form spirals, whereas amylopectin, it is contained 80%, it is made up of branched chains of alpha glucose units. Structure of starch here, you can see alpha glucose. You can see one sugar molecule, another sugar molecule linked to alpha 1, 4 glycosidic bond linkage, and whereas amylopectin, you can see here alpha 1, 4 or 1, 6 glycosidic bond linkage. Coming to dextran, it is a complex branched glucan composed of various lengths of chains. It forms from 3 to 2000 kilodalton. It is used medicinal purpose. It can use for anti-platelet, to reduce blood viscosity, and as a volume expander in hypovolemium. It is synthesized from sucrose by certain lactic acid bacteria. And coming to dental plaque is rich in dextrans. The straight chain consists of alpha 1, 6 glycosidic linkages between glucose molecules, while branches begin from alpha 1, 3 linkages. You can see here dextran structure, and this structure, the sugar molecule linked to alpha 1, 6 and alpha 1, 3 linkages. And coming to cellulose, cellulose is a non-sugar molecule and homopolysaccharides composed of glucose sugar units, polymer of beta glucose, and each monomer is inverted and as a consequence for its properties. So glucose units are linked by alpha 1, 4 glycosidic bond linkage. It is not hydrolyzed. It is hydrolyzed by only with acids and alcoholic solution. It is main consistent of cell walls of plants. It actually contains more in the cell walls of the plant. It occurs lignin and cotton. Its molecular weight is very high. It contains more than 12,500 glucose units per molecule of this cellulose. Coming to glycoproteins. When you talk about the glycoproteins, you can see here glycoproteins are proteins. It is nothing but the proteins that actually contain oligosaccharides chains. And it is actually covalent attached to the polypeptide side chains. The carbohydrate is attached to the protein in a co-translational or post-translational modification. This whole process we call glycose glycosylation. The glycoproteins are important integral membrane proteins where they play an important role in cell-cell interactions. There are two types of glycosylation. So, nitrogen glycosylation, the addition of sugar chains can happen at the amide nitrogen on the side chain of the aspargine. Aspargine is nothing but amino acids. So, nitrogen glycosylation, actually addition to the amide nitrogen, okay, on the side chain of the aspargine. Oxygen glycosylation, the addition of sugar chains can happen on the hydroxyl oxygen on the side chain of hydroxylysine, hydroxyproline, serine, threonine. So this is glycoproteins. You can see here linked to oxygen and then nitrogen linked. You can see here these are amino acids linked to oxygen. Okay, aspargine. Aspargine is linked to N-linked. Okay, and then you can see oxygen linked. Okay, serine, threonine. This is amino acids. So these are the oxygen linked glycoproteins and then nitrogen linked glycoproteins. And then glycoproteins are very important for white blood cell regeneration, especially in the mammals. Molecules such as antibodies, immunoglobulins, which interact directly with the antigens are glycoproteins. Molecules of the major histocompatibility complex, which are expressed on the surface of the cells and interact with the T cells as a part of the adaptive immune response. Glycoproteins 2B and 3A, an integrin found on platelets that is required for normal platelet aggregation and adherence to the endothelium cells. So, components of the zona pellucidea which surrounds the victim and it is important for sperm egg interaction. Glycoaminoglycans, GAGs, usually we call short term GAGs, glycosaminoglycans are long unbranched polysaccharides consist of repeating disaccharide units. The repeating unit consists of amino sugar unit. So, amino acetyl glucosamine or N-acetyl amino galactosamine along with a urinic sugar or galactose. So, GAGs are polar and attract to water. They are very attracted to the water. They are therefore useful to the body as a lubricant or as GAGs have a high degrees of heterogeneity with a regards to molecular weight, mass, disaccharide construction and sulfylation. Based on the core disaccharide structures, GAGs are classified into different groups. So the examples heparin and then dermethane sulfate and keratin sulfate and hyaluronic acid. So these are actually these molecules. It is in the Golgi operators with oxygen linked and nitrogen linked glycosylations as a post-translational modifications. Hyaluronic acid is synthesized by integral membrane synthesis, which immediately segregate the dynamically enlonged disaccharide chain. So this is the structure of GAGs. These are the examples of GAGs: heparin, keratin sulfates, and then hyaluronic acids. So this is, as I mentioned, the 4-1 we discussed here. And the functions: heparin is administered as an anticoagulant. Heparin usually we use anticoagulant. Heparin sulfate has a numerous biological properties and functions, including cell adhesion, regulation of cell growth and proliferation, development of process, cell surface binding of lipoprotein, lipase, and other proteins, angiogenesis, viral invasion, and tumor metastasis. Dermethane sulfate interact with the fibroblast growth factors FAGF2, FAGF7 have been implicated in cellular proliferation and bone repair. Keratin sulfates helps in the maintenance of the tissue hydration. Hyaluronic acid is a major component of synovial tissues and fluid as well as other soft tissues and endows their environments with remarkable rheological properties. Lectins are the carbohydrates binding proteins, macromolecules that are highly specific for sugar moieties. Plant lectins are also known as the phytohemagglutinin were noticed for their particular high specificity for the foreign glycoconjugates. Examples, those of fungi, invertebrates, animals used in biomedicine. These lectins are used in biomedical applications, biomedicine for blood cell testing, fractionation. Lectins perform reionization on the cellular and molecular level and play numerous roles in biological reionization phenomena involving cells, carbohydrates, and then proteins. Lectins mediate attachment and binding of bacteria and viruses to their intended targets. For example, it is hypothesized that some hepatitis C viral glycoproteins attach to C-type lectins on the host cell surface, liver cells for infection. So aneurysm is a carbon one in the cell cycle structure. Is called anomeric carbon atom. It is a asymmetric carbon atom. If the hydroxy at carbon 1 position, if the hydroxyl group on the carbon 1 position is to the right side, it is called alpha form. If the hydroxyl group at the C1 is to the left side, it is called beta form, that is B form. So this is the structure of aneurysm. So you can see here alpha D glucose and then beta D glucose. Alpha and beta forms of sugars are called anomers. You can see the left side and then right side. You can see the carbon 1 position hydroxyl group. If it is the right side, alpha D glucose. If it is the hydroxyl left side, the carbon position 1. So that is beta D glucose. Alpha and beta form sugars are called anomers. This is a type of stereoisomerism. Coming to isomerism, isomerisms are chemical compounds having the same molecular formula but different structural or stereochemical formula. Functional group isomerism. Example, aldose and ketose isomerism. Glucose and fructose are aldose, ketose isomers. So glyceraldehyde examples and then dihydroxyacetone are aldose ketose isomers. Beta rotation, when actually happen alpha and beta in interconversion of alpha and beta anomers, the alpha and beta anomers of carbohydrates are typically stable solids. In aqueous solution, they are quickly equilibrate to an equilibrium mixture of the two forms. For example, in aqueous solution, glucose exists as a mixture of 36% alpha and 64% beta form. So when the greater than 99% of the pyranose forms exist in the solution. So this is actually today we had a more discussion about this polysaccharides. So we'll learn more about the types of the polysaccharides and then structure of glucose and then starch and then cellulose. And then we have discussed GAGs and then glycoproteins and then isomerism and then aneurysm. So these are, we'll learn a lot more about this one, and then hope you will read this one, and we will see next class we will discuss more about the another topic. So thank you all.